Thank you very much for that introduction. Do not ask me about my PhD mentor, only in the break, okay? Very good. So my intellectual history, as you can tell, is somewhat mixed. I studied biochemistry originally and was always intrigued, even at that point, by the intersection of biological systems and math or computation or modeling. In fact, one of my first papers is on modeling a genetic regulatory circuit with differential equations and chaos, actually, thrown in the mid-'80s. So I have sort of taken that routine into my adult professional life as well, where I'm still very much interested in the intersection of theory and experiment, data and computation. And I am now at the current stage of my career really approaching the brain from a network perspective. And I want to tell you in detail what I mean by network. I don't mean network in the classic sort of sense of interconnected neurons or whatever, but really in a network science sense as a model of a complex system. So I will come to that in a second. So I see networks as they are seen by many in the field now, as representations of a complex system that we've taken apart and divided up into nodes and edges, elements and interactions. The relationship of the nodes to each other as defined by the edges is then typically called the network topology. And analyses and models of such networks have made a really tremendous impact, I think, across very different scientific disciplines, including the social sciences, where some of the thinking originated in the 20th century, and then also more recently in technology, internet, of course, and also biological systems. And I'll show you a couple of renderings here, visualizations of complex networks from the social sciences. On the left, a political blog network. You can imagine what the two colors, red and blue, refer to, and it's visualizing kind of the tribal nature of our interactions in the political sphere these days. On the right, interactions among protein molecules, which are the nodes in this rendering, and physical interactions among them are the edges. And there's thriving fields out there in social sciences, technology, and biology that try to make sense of these network architectures and understand them from the perspective of their respective disciplines. And I think that the case I want to make here today is that network science, these kinds of approaches, the term network applied in this sense, can make an important contribution to understanding how brains are structured and how they function. So first, a little bit of an introduction into how I define brain networks and where they come from, how we construct those types of things. Then a survey of brain connectivity, mostly anatomy, across different species, model organisms, including non-human primates. Then turning to humans and neuroimaging approaches to constructing networks and the architectures that we've learned about in the last decade or so. And if I get to it, some computational models of so-called functional connectivity. So a few words about the importance of network science in brain. It's really increasing in the past few years, in part driven by the availability of data. I think this diagram here may look familiar to you. In fact, I think Brian had a version of it in his talk. It's deliberately modeled on an earlier diagram by Sejnowski, showing here, however, that across different spatial and temporal scales, a lot of the data that we now have access to or acquire in neuroscience settings, all the way from behavioral experiments down to molecules, really represent interactions or relational data, represent larger systems that we're sampling, and what we're often interested in is binding events, connections, coherence or correlation, coincidences, relational data among the elements that we harvest and that we then represent and try to model and understand. That's where the data-driven aspect of the ascent of networks in neuroscience, I think, comes from. That's how it's fed, and that's why understanding networks and network terminology and network methodology across these different scales is becoming increasingly important, I think, in our field. Different way of looking at it is this way, sort of reading the slide from the top down. We have molecular screening techniques. We have anatomical mapping that's getting finer and finer down to the EM level. We're having high-density recordings in electrophysiology and imaging, and we also have high throughput, high-volume behavioral tracking increasingly in people and in animals. All of those data percolate down here. Quite often now people construct networks from these data, whether it's networks of anatomical connections or functional interactions or behavioral interactions. Those create network representations or networks which we can then analyze using a common toolkit of methodologies and measures and metrics that are delivered to us by our colleagues in network science. And then, of course, the cycle begins again. Network science applications generate predictions that have to be tested experimentally, and the whole cycle starts over. So there's a lot to digest, a lot to do here, and we and others are now increasingly referring to this whole enterprise as network neuroscience, where essentially this field is sort of positioned in the intersection between empirical brain science on one side, computational neuroscience, and network science as well. The network measures that we have at our disposal, some of them are quite simple, such as the degree, which is just the number of edges that are attached to a given network element or node. Others are quite complex, such as modules and rich clubs. So the node degree is the simplest one. I mentioned it. Clustering coefficient quantifies the cliquishness of a neighborhood of a given network element, quantifying essentially the ratio of the existing connections between a node's neighbors and the total number of possible such connections. Motifs are subgraphs into which a network can be decomposed. We can compile statistics on those subgraphs and compare them to null models, etc. A very important concept in graphs and networks are paths, because usually graphs, biological networks, are quite sparsely connected, and it stands to reason that it is important to not only quantify direct interactions between network elements, but also indirect interactions that travel through intermediate steps. In this case, node A and node B cannot talk to each other directly, but can only talk to each other through at least two intermediate nodes on the way. That path length of 3 is the minimal length that these two elements can communicate through, and that concept of distance, expressed here as a topological distance of number of steps, not metric, is an important ingredient in many of the metrics and algorithms that I'll talk to later. At the bottom then, a very important technique in network science is decomposition of larger networks into clusters or communities or modules, as they're called. I don't mean modules in a cognitive science sense, but in a sense simply as highly or densely connected subparts of a larger network. There are many algorithms, many approaches for how to do this, and I'll refer to a few of these later. An important corollary of that is that highlighting once we have a partition of a larger network into compact clusters or subunits, it highlights the importance of certain nodes shown here in black, one of the examples shown here in black, that are critical for maintaining interconnectedness among these modules, and informally, somewhat informally, these are often called hub nodes. Obviously, if that node were perturbed or if it was lost entirely from the network, these two communities would now lose entirely the capacity to communicate with each other. On the bottom right, another concept that I'll refer to quite frequently is, once we've identified highly connected hub nodes that are linking communities to each other, we can also quantify how dense is the connectivity amongst them. That is a measure or metric that is called in social sciences the so-called rich club. Rich does not refer to money here, but to number of connections that are attached to a given node. And the clubbiness is that these nodes also are connected amongst each other with a density of connections greater than expected by chance. It turns out that rich club architecture is a theme that runs throughout my talk, because it is actually an attribute that we see a lot in networks across different species. So particularly the modularity, community detection, and rich club architecture is something that I will refer to throughout my talk. Now, before we can start applying network tools and methodologies to our data, we first have to construct networks from empirical recordings or observations of anatomy, and that is an arduous process, a process that consumes entire careers and PhD-ships and postdoc fellowships and so forth. It's actually very difficult to do in neuroscience. Data is often noisy. It needs to be pre-processed. It needs to be mapped onto standard coordinate frames in certain ways that are time-consuming and sometimes, in some cases, not even standardized. But we can do it on almost any anatomical or functional data set that we can get our hands on. That's why I'm showing at the top examples of nervous systems, not just human in the middle, but also nematode, Drosophila, the rodent brain, and the non-human primate. In this diagram here, let me focus on the human brain for just a second. The way we construct networks from human brain imaging data is typically by taking, let's say, a gray matter surface and breaking it down into parcels or regions according to some anatomical or other template. Unfortunately, that process is there's not universal agreement among practitioners in the field as to what the correct parcellation should be. So there's many different alternative possibilities that we can pursue. But nevertheless, once we have done that, the parcels become our nodes in our network, and essentially we're now constructing a lookup table of what's connected to what using anatomical observations, which in an animal model might come from fluorescent imaging or radioactive labeling or tracer experiments, and in humans typically comes from diffusion imaging and tractography. The output of that is a matrix shown on the lower left of weighted associations between nodes which describe the strength or magnitude of anatomical connections, and the complete picture of that for a nervous system at a given resolution is what we proposed to call, some 15 years ago now almost, a connectome. And it's an ohm in the sense that it is foundational for understanding how, in my opinion, for understanding how nervous systems function. And similarly, as genomes are foundational for the rest of biology, then they don't tell us all the answers, okay? And in fact, they raise many questions about how we can conceptualize our understanding of connectomics on the one side and our understanding of brain dynamics and behavior on the other. There's not a simple one-to-one relationship between the two. Nevertheless, I maintain that connectomes are foundational ingredients for understanding how nervous systems operate. On the right is a different workflow. We're recording, in this case, from, let's say, sensors on the brains, on the surface of the brain or the scalp, or we're recording from voxels inside the brain, let's say bolt signals. We're getting what I like to call wiggly lines, which are the ups and downs of a voltage or a bolt response, which can then be processed using time series techniques into something as simple as a correlation matrix, which you see down below, or more complex relationships such as Granger causality or Bayesian networks or what have you that capture more closely capture causal relationships and directed relationships among different elements. Once again, this is done in a parcellation, either it's sensor space or it's a space of brain regions. It gives us another matrix of relationships that we can also conceptualize as a network. Now we call it a functional network because it has to do with time series. And we can actually, if we are working in humans particularly, where we can acquire these two types of data in register with each other, we can now compare one to the other and have make interesting studies about how they might be related. I'll say more about this later on. Before I go on, I want to stress there are some commonalities between structural and functional connectivity, but there's also some important differences. Yes, both are networks, both are displaying things like modular organization, community structure, but there are many differences. Structural connectivity is physical in nature. It's real. Connection either is there or it isn't there. You can point to it. It is sparse typically, sparse network. While there may be many synapses and many projections, ultimately, in principle, at least they're a fully enumerable set. And changes in these connections occur, yes, across time, but we think on a fairly slow time scale, at least with respect to the comparison to functional connectivity. Functional connectivity is much more dynamic, statistical in nature. It's only a statement about the statistical dependence or correlation or coherence between time series, and there's no statement here about a physical connection, and it can take many different configurations. It can be measured in many different ways. And I would add that many tools from network science should not and cannot be applied to functional connectivity in the same way as they can be applied to something which is more physical and material in nature. Actually, I had a discussion about this just before the talk with one of the students here. A lot of tools that we routinely apply to the analysis of brain networks that are structural really should not be or cannot be appropriately interpreted with functional connectivity. One thing that can be fully interpreted is modularity, and I will come back to that in the remaining part of the talk. Here's a few stories about where we've been in some ways as a field and in some of my own studies across multiple different species. We now have in our possession a handful of connectomes, or connectome drafts at least, acquired with a variety of methodologies from electron microscopy all the way to imaging from a handful of species. And so that allows us to perform comparative analyses. What are some of the common principles that we encounter in these connection diagrams? What are some of the recurrent themes and some of the differences perhaps between different species at the level of network features and attributes? The field got started some 30 years ago, or more than that, with the first reconstruction of a full neuronal wiring diagram in C. elegans, about 300 neurons altogether. And despite the fact that that connection data set has been with us for 3 decades, there continue to be new studies carried out even today that apply new tools, new ways of looking at the architecture of this particular connectome. It is a seemingly simple organism of only 300, the nervous system of which is only 300 neurons. I hasten to add that while people sometimes refer to it as simple or even primitive or whatever, do not insult the worm. It is a capable organism that's been living quite successfully for millions of years in the ground, below us probably at this point. And it has quite remarkable behavioral capacities and sensory and motor capabilities. So I feel like this nervous system, even though it's quite small, quite compact, has a lot of secrets still to disclose to us, even though we have a full diagram, a connectome in front of us. Just a couple of things that came out of recent reanalyses of this. Well, it has modules. There's a modular organization. There are communities of neurons that are more densely connected in this network diagram. It has high clustering, high cliquishness. Neurons that are connected to each other often have common neighbors. And there's a short path length overall, information being allowed to flow relatively freely through this network, even though it is 300 neurons wide. And highly connected nodes, highly connected neurons with many connections, of which there are a few, also tend to be connected amongst each other, which is, of course, that rich club property I mentioned earlier. These are properties that we find even in this very simple organism, in even this very simple, compact nervous system. On the invertebrate side, I think the greatest promise right now for delivering more complex and I think quite soon single neuron connectome data is Drosophila, about which, of course, we know a huge amount in terms of its behavior and its genetics. In our first exploratory study with Anshin Xiang in Taiwan, they labeled individual neurons, fluorescently labeled individual neurons in the Drosophila brain, essentially imaged them one by one, converted those images into a common spatial template, and reconstructed from that a summary diagram, as it were, of connection density, a directed matrix of connection densities across the Drosophila brain, 49 or 47 local processing units, as they call them. And we and others have looked at this diagram since. One of the prominent features of it is, again, modularity, existence of densely interconnected communities that are interconnected by hub nodes, which in turn are also interconnected amongst each other, something that you've just heard about C. elegans. The mouse, of course, I'll just very briefly mention the pioneering work carried out by the Allen Institute, and kudos to them for making all of those data publicly available. This enables people like Mika Rubinov and Ed Bullmore here to do an analysis of those data in terms of network features and network architecture. Once again, a prominent aspect of the architecture is its modular construction with interconnecting hubs that are interconnected amongst each other to form a rich club. Regions that are members of the rich club are orbitofrontal, anterior cingulate, some thalamic regions, some striatal regions that are interconnecting these different modules to each other. In my own work, I've been, over the last several years, been fortunate to work with Larry Swanson at USC, who is, of course, an extremely well-known neuroanatomist. His organism of choice is the rat, and he conducts the following type series of studies now, and I've been working with him for a number of years, looking at different subdivisions of the rat brain, mining the literature, published literature of connection reports in the histological tract tracing literature, then compiling essentially big spreadsheets of what's connected to what in a given postulation Each of the elements in these matrices that you see in the next several slides reports on the existence and strength of a projection, directed projection between two brain regions of the rat. And many of the entries in these matrices actually have behind them not just a single connection report, but actually multiple reports in literature. So there's a lot of published literature out there, and mining that literature, converting that into network representations is something that Larry does with extraordinary precision. We started out some years ago in the rat's cerebral cortex, one hemisphere, and here's this is a rendering of the connection matrix with the modules highlighted. There is modular architecture, high clustering, short path length, and you guessed it by now, also rich club organization. Interestingly, those modules are spatially compact when you reflect them back onto what Larry calls the flat map, the flat map preserving spatial neighborhood relationships or adjacencies along the cortical sheet. Those modules actually are occupying contiguous territories. This tells you something about the relation between spatial embedding of brain regions and their propensity for being connected, a theme that I will come back to in just a second. We have...
The ordering you've got in matrix,
yep.
That's not obviously arbitrary.
No, it's not. It's not arbitrary, even though the log scale is not perfect for the rendering here. It is the result of an algorithm that's been applied to detect modules. The algorithm comes from network science. It's called a Louvain algorithm. It's got a metric behind it that we optimize. That whole part is actually technically quite complicated, but I'm skipping it here. But essentially, I've done nothing to the data. I've just reshuffled the rows and columns of the matrix. It's in some ways a visualization trick, but one that also gives me membership for each brain region into a module, and I can then compute subsequent features of the network that relate to the partition and then going forward. So that's the power of this. I have colleagues in my university that have built their entire careers on building capable algorithms for doing this on very large data sets. So this is actually an active field in computer science. Well, here we have the spatial embedding of these modules. Here's a subsequent follow-up. These are two cortical hemispheres now, right cerebral cortex, plus the commissural connections thrown in. The commissural connections are on the upper right and lower left quadrants of the matrix. You can perhaps see that prominent diagonal streak going through that part of the matrix. Those are homotopic connections that interconnect corresponding brain regions across the cortical midline. And there was very little data available until the study as to how commissural connections are actually laid out in the mammalian brain. And I think we were able to shed a little bit of light on this with this study of the rat cerebral cortex. Some of the things that we found, not all connections are homotopically, not all brain regions are homotopically connected. Only about two-thirds of them are. In fact, the entire set of heterotopic commissural connections is quite sparse. Only about 3 or I remember something like 6 or 7% of them exist. Commissural connections are an exact subset of association connections occurring on the exact same, on the same side in each cortical hemisphere. So there are no commissural connections that are new or non-existent on the same side. And they tend to be quite a bit weaker in strength than the corresponding association connections. Notice that when we decompose the network into, in this case, 3 modules on each side, the interconnections among these modules are quite symmetric. That's an arrangement that we do not see in the basal ganglia, which was our third iteration of this. The striatum and the pallium here, the 2 major subdivisions of the basal ganglia in the rat, and their interconnections. This is the primary data set from which all the analyses were derived. If we perform modularity analysis on this data, we find nested modules at different spatial scales that represent different types of orderings in striatal and pallidal circuitry. These, at the top level of this ordering, those modules that we find, 4 of them, have characteristic associations with particular types of behavior. So their outputs, their relationships, for instance, with the hypothalamus, predict that they are engaged in particular different types of behaviors. When we look at the summary diagram of the mean connectivity between these modules, it looks strikingly different than cerebral cortex, much more asymmetric, much more loop-like, much more unidirectional. And that's a type of coupling that seems to be unique to this particular structure. The last iteration of this is work currently in progress. Our next iteration, we're starting to call it a franchise now, is to take the cerebral cortex, 2 hemispheres, and the basal ganglia, 2 hemispheres, put them together with all the interconnections as well. This is now a 244 region matrix, and it comprises almost in its entirety the rat end brain, the telencephalon. And this is our module ordering that we've derived, in this case using a very state-of-the-art multi-resolution consensus clustering algorithm, which was just recently published by my colleagues at IU. Here's what this algorithm delivers. It delivers to you not just a single modular arrangement, 4 modules shown here, but it also shows you the nested hierarchy of modules within modules. This is a feature that's been underappreciated in these analyses so far, also by us. These data sets of connectivity can be divided into modules, yes, but there's actually many different ways that we can do it at different spatial scales. And this matrix here, the so-called co-classification matrix, quantifies for each pair of nodes the likelihood or the probability with which they are associated with each other across multiple spatial scales. The lighter the color, the whiter it is, the more densely they are connected and associated with each other in terms of modular structure, and if it's black, there's no association at all. So this allows us now to form a fully multi-scale picture of how modules are arranged in this type of architecture, and that's been something that I've been very keen to do also in other systems. We're having work in progress also in human brain. Okay, a quick word about the macaque, the non-human primate. This is actually work that preceded the rat work by a few years. And by now, I sound like a broken record. Yes, we can divide the macaque cortex. We and others can divide the macaque cortex into modules and interconnecting hub regions that are also interconnected amongst each other quite densely. The members of the so-called rich club here are regions such as area 9, 46, and so forth. These are orbitofrontal, prefrontal regions, cingulate cortex, parts of the temporal lobe, parts of the medial parietal cortex. These are regions that, if you actually plot them onto the macaque surface, they're quite well separated from each other. They're far apart from each other, and yet they are densely connected. They are themselves rich in connections. They have many connections to many other brain regions in the macaque, but they're also densely connected amongst each other. And that feature of dense interconnectivity of rich club regions is one that I think is important because it signals that these connections might actually be of adaptive value. Why do I say that? I'm going to unpack that in just a second. Just to summarize now, we have gone through a number of different species, from invertebrates to non-human primates, and we found a bunch of features here, not just in our own work, but work from other labs as well, that point at a common set of properties. I want to underscore that these properties emerge from network data sets that have been constructed from very different empirical techniques, electron microscopy, light microscopy, track tracing, as well as non-invasive imaging. So none of them can be viewed as an artifact of a particular experimental approach. Broad degree distributions with some nodes or neurons having many more connections than others, log normal profiles of connection densities, specific connection profiles, each node having its own what we call connection fingerprint of inputs and outputs, high clustering and short path length overall, the existence of modules that are interconnected, and the tendency of highly connected neurons or nodes to also be densely connected amongst each other in the form of a core or a central club or clique. Now you might say, what drives these common properties? After all, C. elegans is quite different, we would think, from a non-human primate, or as you'll see in a moment, from the human brain. What drives these common principles of organization that we have observed? And here it's important to keep in mind that anatomical networks are physical. They are embedded in space, they're geometric objects, and they are subject to constraints, therefore, of volume, wiring length, and also energy consumption. And so a lot of the features that we've seen so far, we think, are due to, at least in part, the requirement, absolute requirement through evolution of keeping the brain within those design specifications. You know, as I sometimes flippantly like to say, if your brain does not fit between your ears, you are not going to have any offspring, okay? This is a constraint that cannot be subverted. And so many studies have shown, in fact, that idea goes back all the way to Cajal himself in the 19th century, that conservation of material and energy is one of the principal constraints of how neurons are structured, how they're organized. We're seeing distance dependence in virtually all systems people have looked at, all the way through to C. elegans. We're seeing that real brain networks tend to be almost minimally, but not quite minimally wired, and they're not quite as important. There are always those additional features we see that really shouldn't be there. If the brain just wanted to be cheap, okay, if all that mattered was economy, keeping the wiring short, then some features like long-distance connections between rich club nodes should not exist. So there's a wrinkle to this. On the one side, yes, wiring minimization, and many people have talked about this for decades, if not over a century. There's also another constraint, which is the brain also has to perform. It has to arguably perform its function in the context of adaptive behavior and perhaps integration of information. And for that, the brain has to remain connected fully, and it also has to move information around, or be able to, at least in principle, from one end to the other with relatively little cost in terms of length of time and number of steps. So we think that the emergence of these universal properties of brain networks, modules, cost-preserving, and hubs, which are costly because they have long-distance projections, these are for us now two pillars, not the only ones in the end, I'm sure, but for now, two starting points to look at design principles, okay? Just to drive this home in a different cartoonish setting. We're thinking of the brain, the way we actually encounter it now, as a design compromise or a trade-off between two tendencies that have to be balanced. On the one side, the tendency is to keep connections short. The brain has to preserve cost or lower cost. But if we let it go too far to that side of the equation, we're getting a brain that looks like a lattice network, which you see here on the left. And that's very cheap to make, but it is also very difficult to get information from one end to the other because there are many steps involved. Instead, perhaps strangely, networks with random topology can move information around at blistering speed, but are extremely costly to wire up. Somewhere in between is where we encounter actual brain architectures that have elements of both regularity or lattice-type topology, nearest neighbors, connections remaining short, but then on top of that, a sprinkle, if you wish, an investment that the brain has made in long-distance projections that create shortcuts and allow brain systems to interact with each other over long distances relatively directly. So that's the kind of framework we are working with. We have not just proposed it as a sort of philosophical idea, but we have also built implementations, generative models, multi-objective optimization algorithms that actually look at this from various perspectives, and that's a line of work that I would love to talk about more, but I can't because I'm going to turn over to the human brain now and leave this track where it stands. For me, a very sobering thought for me was that we think of our brains perhaps sometimes as highly optimal or highly optimized, but these constraints of material, volume, wiring length, and also energy consumption place very narrow sort of design limits on what brains can look like. The brains we actually encounter, the ones in this room, may actually be quite suboptimal. Not the ones in this room. They mine included. Okay, may actually be from a purely design perspective quite suboptimal, because in the physical realm of actually building these things in the real world and fitting into the skull and so forth, brain architectures are quite constrained. I think that it is this point that actually is a real point of departure to artificial neural networks. I don't know if Adrian will talk about this, but I have a hunch that a lot of the architectures that are now being explored in deep learning and multi-layer AI configurations are not subject to any of those constraints of geometry and space and certainly not energy, and perhaps for that reason have a totally different part of the morphospace, as we call it, to explore, while we are stuck over here. We can't get out of the hole that we're in, because our brains can only be so big and can only consume so much ATP. I'll leave this whole line of thought aside and turn over to the human brain and what we know about it. Not talked about it yet. When I first got started, in neuroscience, my interest was sparked by medical textbooks and by illustrations such as this from an anatomical atlas. This is actually a photograph of a human brain that's been dissected, post-mortem, of course. And you can see these tracts of what we call white matter fibers or white matter connections that travel through the interior of the brain, essentially big bundles of wires that connect brain regions amongst each other. You can immediately appreciate that dissecting postmodern human brains is kind of a difficult job. Not a lot of subjects out there volunteering, and you can't actually go back and interrogate these subjects in terms of their cognition and behavior. What we really needed was a non-invasive tool to actually allow us to perform this mapping with people being alive and well. And about 15 years ago or so, we got this technique available to us, diffusion imaging interactography, which allows to computationally infer, build a model essentially of the putative trajectories of white matter architecture in the living brain. I underscored it's a process of inference. It's not a direct observation. It's not a dissection. It's not a stain. It's none of that sort. It is an inference on the basis of a complex diffusion signal, and as such is subject to false positives and false negatives. And we have to be honest about this. This technique has its issues, let's say. Nevertheless, it is extraordinarily powerful when applied properly in allowing us to have access to white matter anatomy in individuals, in people, where we can study the variation among them and the relation to cognition and behavior. So it's been an enormous step forward, I think, even though the technique often gets criticized, I think, somewhat unfairly. We worked with Patrick Hagman in an initial study about 10 years ago now to map the white matter architecture in a massive cohort of 5 individuals. We could never get this paper published today, never again, 5 individuals. And we built a network diagram from that white matter reconstruction, of course, and performed network analysis on modeling of that diagram, and found features that have since been found again and again and again in other cohorts. So our cohort of 5 was maybe very small, but it turned out it actually replicated. All of these features are now, I think, established as hallmarks of how human connectomes are organized. And I would hasten to add that in the meantime, last decade or so, the balance has shifted from just descriptive analyses of sort of measuring things like clustering and modules and so forth in such data sets to building generative models, building models that ask the question why, or what are the principles or ingredients that are behind the emergence of these features that we observe. What gives rise to modules? What growth processes? What pruning processes? What variations among individuals can we account for using relatively simple generative models? And there's been a lot of progress in the last couple of years in this area. I think it's the way the field is shifting now, away from description to more models that are generative and predictive in nature. Coming back to the Rich Club one more time, this is actually strangely enough, it was the human brain where we first encountered this property. Just want to unpack just a little bit more why we care so much about it, or I care so much about it. On the left, you see a simple schematic of a modular network where you have communities of nodes that are densely connected, and then we have a few of these black nodes there which have many connections, but also have important connections that go across modular boundaries. And if everything would just look like that, that would be kind of cool, but there's something missing, and that's the ingredient in the middle, a slight addition of additional connections between these hub nodes that perhaps is a little bit above chance, what we expect just by chance based on their degree. And that's the Rich Club architecture. It is a non-trivial aspect of complex networks. It's not found in all biological networks. For example, protein molecules, highly connected proteins that have many binding partners in the cell typically are not binding to each other, for example. They stay away from each other. It's a different architecture. We found it interesting to look at Rich Club architecture because of existing theories in cognitive science and psychology about integration information and what might be substrates of that. going back several decades, including the workspace theory of Stan Dehaene for consciousness. And we find that such a physical infrastructure of densely interconnected multimodal association region, as it turns out in just a second, would be a good substrate for integrating information and sharing information quite easily within such a network and integrating it. Working with Martin Vandenheuvel at the time, we looked at two cohorts. Actually, the study had a replication cohort built in. Diffusion imaging spectrography, reconstruction of connections across the human brain. The key graph is the one at the top in red, which signifies the increased density of connections among high-degree nodes with many connections, over that expected by chance from a rewiring null model. Down below, you see kind of, perhaps you can tell roughly where these rich club regions are located. They're dispersed across the brain. Prominent regions are in the cingulate cortex, posterior parietal cortex, and in the insula. And if we try to plot communication paths through this network, we ask the question, if we want to go from A to B anywhere in the brain, how often do we have to travel through that red portion of the network, the rich club itself? Despite the fact that the rich club is only about 10% of the infrastructure of the brain in terms of nodes and connections, 90% of all shortest paths in the brain have to travel through some form of rich club architecture. So it's actually we call it sometimes colloquially the highway system. Why? Because if we travel, especially across over long distances, transportation networks are laid out in such a way that people have access to these long distance highways, and these are sort of our rich club pathways that we have here. Final point about the human brain is that a lot of interest is motivated by our lack of understanding of neurological and mental disorders. And one of the common themes in the literature now, currently writing a review article, another one on these, on what's called disease connectomics now. One of the underlying themes is that many disorders with very different etiologies, very different labels, appear to be starting in what we might call hub nodes, highly connected parts of the brain that are not only rich in connections but also interconnected amongst each other. The diagram on the left is from a meta-analysis by Nick Crossley, who examined a total of, I think, over 20,000 T1 scans of people with various disorders, 20 or 30 different disorders. You see them listed here. And he looked at, using T1, which is an anatomical scanning modality, he used that to identify where signs of atrophy occurred first in these individuals. And while this is not true for all disorders, but many of them, statistically many of them, the trouble sort of starts with more highly connected parts of the brain. And that's an interesting lead that's been pursued along many different avenues. We're working on data with our colleagues in Indianapolis at the Alzheimer's Center in dementia, where we also feel that that is a potential pathophysiological mechanism beginning of the disorder in hub regions that are highly connected, which are then spreading potentially misfolded tau proteins. We're looking at this with tau PET and other imaging modalities with them now. Finally, a few words about functional connectivity. So far, I've only talked to you about anatomy. I actually am a very structural thinker, okay? Part of the reason why I was interested in biochemistry as an undergraduate was because I was fascinated by protein molecules and the history of biochemistry had just gone through its sort of golden phase, where if you had solved a big molecule, you got a Nobel Prize, okay? And there was a reason for that, because these molecular structures were extraordinarily informative about how these proteins actually operated, what kind of environment they resided in, how they interacted with their substrates, if they were enzymes, how they bound to each other. And for me, this was fascinating. So it's fascinating also because I feel like evolution, if it wants to do anything and change something, it can't change function directly. It has to work through structure. It has to do something with a protein side chain, has to swap out an amino acid for another one, or create something in the anatomy of an organism that enables a different type of behavior. But it has to be structural, ultimately. So I'm a very structural thinker. I feel structure, and the connectome for that reason, is primary in importance. But nevertheless, we are interested, certainly, in what occurs when the brain does become active And I'm showing here the key question, which is how do we get from something which is an anatomical skeleton, the connectome on the left, the stuff we've been talking about so far, to something which is dynamic, changing, adaptive, responsive, and visually engrossing as this movie of a resting brain in an MR scanner, which is shown here on the right. This is a revelation for a different reason. It's actually a brain at rest. There's no task being performed here other than staying awake and don't move and so forth. If we put a cognitive activation on this in this diagram here, it wouldn't show, we wouldn't notice it. It would have the same amplitude as any of these bumps that you see of activity that occur seemingly in a random way in this resting brain. Now this connectivity, this pattern is not random, it turns out. Resting state functional connectivity over the last 10 years has expanded tremendously precisely because the pattern that you see, or perhaps don't see in this movie, but you'll see in a moment, actually is extraordinarily informative about individual differences in the human population and also disease states. Here's how we get to seeing those individual differences. We take a movie like this, and we take individual frames, we cut up the movie into frames, and we cross-correlate these frames across time. It's as simple as a cross-correlation. I'm embarrassed to say it every time, but it is simply a Pearson correlation, actually a partial correlation, but nevertheless. And that gives us a functional connectivity matrix of the kind you see here on the right, a fairly arbitrary example. This has already been clustered, or module detection has already been performed, so I've already arranged my nodes and edges so the arrangement makes some sense to us. And the blocks that you see along the main diagonal are very reproducibly found across the population. Every one of us in this room, I would bet you $1 million if I had $1 million. Everyone in this room has a default network. Everyone in this room has a visual network, a smartomotor network, a saliency network, a limbic network. These are building blocks that have been discovered in large cohort samples, and the two studies down below by Randy Buckner and by Steve Peterson, and have been reliably reproduced, I think, across many subject cohorts now. What's different among individuals is the degree to which these systems are coherent, the way exactly they are laid out in the anatomy, and the way they interact with each other. There are subtle differences in these interactions that it turns out are really exciting currently in network neuroscience. I'm a computational neuroscientist at heart, so what did we do in this endeavor? We started out very early on looking at spontaneous activity in models, in computational models that were embedded with connectome connectivity as the coupling structure. We started out with in 2007, this precedes the human connectome era. There was no human connectome available to us at the time. This is the Van Essen data set from the 1990s, collation of track tracing reports in the macaque, mostly visual cortex, a binary matrix, what's connected to what in that system, 47 nodes altogether. And we combine that with a biophysical implementation of what neural populations are doing at each node, interacting amongst each other, an excitatory and inhibitory population in particular. Now, the equations we implemented look like they have many free parameters, but that's not the case actually. The only parameter we're allowed to vary is the coupling constant. All the other parameters are fit from empirical data, and we never touch them. So we now have a way to implement an entire good part of the macaque nervous system as a coupled system of ordinary differential equations. This is now a physics problem. We can solve these equations numerically in a simulation. We can lay on top of that a synthetic fMRI mechanism even. This is sort of perverse. We can simulate this at 0.2 millisecond resolution, and then we put a bold function over it. We get bold responses. It's like composing, Mozart composing a symphony and then passing a bandpass filter over it that takes a single note every 2 minutes, every 2 seconds or something. Anyway, this is a system of 141 coupled differential equations, ultimately, 3 at each node, 47 nodes altogether, and the coupling structure is given by the connectome. We get back from that bold time series, except without a scanner. It's much cheaper. And we can then compute things like a functional connectivity matrix from that, which we did here in this diagram. We can do other things. We can perturb the matrix. This is where computational neuroscience becomes an exploratory tool. We can actually look at the impact of lesions in the matrix itself or addition of connections. We can simulate even task states. Oh, this is meant to be a resting state simulation. At the time, we had no empirical data from the macaque monkey. Only years later did we get access to a couple of monkeys that had been measured by Yasushi Miyashita in the University of Tokyo. And we compared our very, very simple, extraordinarily simple, incomplete simulation with the empirical data from macaque monkey and found a healthy relationship between them, despite the fact that no data fitting was carried out as post hoc. This also works in the rat. I won't show you this. At the end, I want to make a couple of points. As a computational neuroscientist, there are sort of two different routes one can take, I think. The route that's taken more often is to take a simple model, such as this so-called neural mass model I just showed you, where each brain region is only represented by two variables, excitatory and inhibitory, sort of like a field model of what goes on in this area. We can take that model, we can unpack it further, add cortical layers, add spiking neurons, add higher temporal resolution, more precision, more free parameters if you wish, more detail. We decided to go in the other direction and try to see how far we could get, at least in modeling functional connectivity, by using much simpler approaches, including analytic approaches from graph theory. This model here was built by my former postdoc, Joaquin Goni, and essentially is a multilinear model linearly combining with 4 free parameters, 4 different graph metrics computed directly on the structural connectivity. The fancy diamond plot here on the left half of the diamond is an empirical recording, a functional connectivity matrix, bold cost correlations in resting state from a set of individuals in the right cortical hemisphere. The diamond part to the right is the model prediction, which is driven by the anatomy and those graph descriptors that we've computed analytically from the anatomy. The model runs in under a second. The neural mass simulations I showed you earlier take on the order of 24 hours to complete, even with modern computing tools. So this is a much, much simpler approach, and we're getting fairly good prediction of, by prediction I now mean building a model based on anatomy, having a few graph metrics on top, linearly combining them. That's the prediction of functional connectivity. Another line of work done with my former postdoc, Bratislav Misic, who is now faculty at McGill, and Rick Bechtel, my former graduate student, is to look at a so-called spreading model, a model where we place a perturbation in the network. We color the perturbation, in this case red and blue, no intention here. We did this unintentionally, but red and blue. And we're letting that perturbation ripple through the network using a simple spreading algorithm that was first devised in social sciences by Mark Granovetter, famous social scientist in the 1970s. The purpose of the algorithm back then was to track the spreading of rumors, innovations, and even unrest in a social setting. We're using that simple spreading algorithm, letting loose our spreaders from given seed points, and then we ask the question, where do the fronts from these two perturbations collide? Think of them as throwing a pebble in the water, another pebble in the water. Where do the ripples from these pedals actually collide in the network? That's, of course, defined by the topology of the structural connections. And we do this for all possible perturbations. We go through all of them. And then we build a heat map as to where the fronts collided in the brain. This is the map shown at the bottom. And interestingly, if we look at those locations and where they occur and how they relate to what we seem to know about functional systems, cognitive systems in the brain, they seem to aggregate more strongly in those systems that are associated classically now with multimodal or higher order behavioral cognitive functions. I think I'm going to skip the final part, and I'll stop here with the next 2 slides and then go to my conclusion. Here's how all of this modeling work and analysis of networks, here's the picture that I have in my mind right now about how all these different threads of investigation hang together. We've written a review article on this just recently on this. As usual, I start from structural connectivity. For me, that's primary. The wiring pattern, what's connected to what, it doesn't determine with certainty what will happen in the brain, but it excludes with certainty many, many alternative possibilities. And that is equally as important, I think, in biological function, excluding a universe of possibilities and then having still a lot of possibilities open that can be taken. So that's the wiring diagram, that's structural connectivity, and for me, that's where it starts. Then we have signaling events within this network, because neurons and brain regions, of course, produce signals that are exchanged and sent down external pathways and get exchanged through synapses. Those signals unfold, barring any major surprise in the future in physiology, for the most part in external pathways, and really ripple through the physical architecture, the connectome itself. And that sets up a fast-changing picture of signals that are traveling through external pathways and synapses, and that's what's meant to be symbolized by the picture in the middle here. And since there are many signals traveling at any given moment in time, you can think of this as a dynamic system in itself. This is an ebb and flow of signaling events that are actually very hidden from us. We actually have no access to these, I think. I can explain why I think that. And for me, that's, however, almost a primary currency of how large brain systems cooperate and work as a whole. Oops. What we do have access to is functional connectivity, but unfortunately, it's a very incomplete picture. All we see in functional connectivity is the effects of those signaling events. We're seeing the signaling events we don't see, but they have effects on the individual elements because they change the statistics of the firing of those elements, which now change the probability distribution functions of how things relate to each other in the brain, right? Certain neurons start firing together, others lose synchrony. It is those types of relationships that we can observe, we can tap into with neural recordings, also with bold recordings, that define functional connectivity, but it's only a statistical picture of relationships that travel kind of through the ether. We've lost the causal picture of what actually goes on in the physical wiring architecture, the signaling events. That's lost. We don't see that. We just see the effects of that. So I feel like very strongly that that middle ground of communication dynamics, the ebb and flow of signaling events themselves, is really a blind spot that we have in our field and that we need to address. And in our field sometimes, this is another term that Carl Friston uses a lot, effective connectivity. It's a little less mysterious than free energy, but I think that is what we do need to drive at in our accounts of how brain circuits and systems give rise ultimately to cognition and behavior. I have gone on enough, and I will cut myself short, even though there is so much more good stuff to come. I will go to the end, to my conclusion. There we are, outlook. So one of the themes, of course, in my talk has been networks, and I've mostly spent my time up in at the systems level of at least systems, perhaps cognitive neuroscience type interactions. And but it's important to realize that for almost any neurobiological phenomenon that we're really interested in, there's networks that shape those phenomena that are not only at the circuit level, but also at the individual neuron level, and even at the level of networks unfolding within neurons themselves in terms of molecular interactions. As I sometimes like to say, it's networks all the way down. And because of the multi-scale interactions of network interactions, this is really a very, very complicated task for us in neuroscience to unravel what is going on in terms of the system's behavior. Because of the ubiquity of networks in neuroscience, and because of the fact that I have nothing to do with my free time, I started a new journal, which is called Network Neuroscience. It's published by MIT Press, open access, of course, and it's designed to really create a community of researchers that range from molecular to whole brain studies across all species, all systems, very much welcoming computational theoretical work as well, to empirical studies, and publishing reviews, perspectives, data papers, methods, and original research articles. We're in the second year of publication now. We're doing extremely well, but I want to no talk of mine would be complete without advertising the opportunity presented by this new venue. So thank you very much for your attention. No, it's a very good question. I've only addressed in this talk here, sort of resting state or spontaneous activity. In a model, it's difficult to talk about resting state, spontaneous activity, type dynamics. But others in the field, of course, have asked, how do these functional connectivity networks change when people are performing mental operations or tasks? In particular, several studies have been carried out with tasks that are kind of ongoing, such as a navigation task, a mental arithmetic task, a working memory task, or something that's not cut up into blocks but kind of has a continuous task structure. And in fact, you're right. Functional connectivity is demonstrably, reproducibly, and characteristically changed in those different task conditions. And it's an ongoing quest to figure out exactly how that maps onto activations. Of course, those we've known for a long time. The field got started on activations, mass univariate testing, and those types of models have been endemic in fMRI for 20 years. The connectivity domain is, for many reasons, more statistically more demanding because you have a lot more to look at now because of the n-square phenomenon of blowing up the number of elements you're looking at. But the short answer is yes, functional connectivity is modulated during task and in ways that are characteristic, and I don't have time to say a lot about it in my talk, though. There's no point in trying to rename things. I've tried. This is something it just doesn't. I know, but it's again Carl. It was Carl actually. I'm not going to blame him for it, but he actually introduced functional connectivity in 1994 as a measure of statistical dependence between otherwise remote neural events, and that's what it is right now. It's neither functional, if you wish, because it does not relate directly to any specific function as in resting state, and connectivity is a stretch because it is a statistical construct and not a physical interaction or a directed causal interaction either. Having said that, though, I don't want to appear that I'm bashing it. I think we have to just look at it with a little bit of what's the right word here? We have to put it in context and not reify it too much. It is an important signature of what the dynamics look like. It is a covariance, a picture of the covariance structure of the system in some ways. But that's not the only way to quantify dynamics, first of all. And it is not one-to-one related to the underlying anatomy. So when you see a strong functional connection between two regions, let's say you place a seed in the anterior singular, anterior insula, and you're seeing other regions appear, it is not necessarily the case that what you're looking at, what you're highlighting now, is a physical pathway that is engaged in that correlation. It can, however, in our models, they are often indirect pathways. So it almost never happens. It can't happen in the brain really that things are so disconnected from each other that it's just a chance correlation. There's dense connectivity, but each observation of a functional connection really is a mixture of potential direct link between these two regions or indirect links, usually many of them, that are superimposed.
But it could also be a third variable, which would be like a global modulator or something.
It could be. In fact, you're absolutely right. In fact, we can never explain, and even our most sophisticated models, we can never explain more than about half the variance. The other half has to come from somewhere else if it's not complete junk. And it most likely is neuromodulation. You're absolutely right. And so global state changes that change effectively the responsiveness of regions locally and thereby modulate correlation strengths. And it could also be subtle modulations in the hemodynamic response, which is known to be regionally specific. We don't have a lot of data in human brain as to how exactly it varies, but we do know that it does. And so that's another mechanism by which a local change in a region, perhaps due to a biochemical or a signaling event, can modulate or alter functional connectivity profiles. But a big part of the equation, I think, is that neural signals have to travel through anatomical pathways. That's the only way they can express their effects on time series on the correlation structure of the system. Yep.
I'm curious if you use the same approach to study the development of networks, so how the structure, connectivity changes over time with age, and what are your kind of surprising findings?
Absolutely. I mean, we have done some studies with lifespan data, which is publicly available through the NKI consortium on the order of 900 individuals from a county in upstate New York, Rockland County, that have been imaged. And we have processed that data and actually have written papers on exactly what you're referring to, that is changes in the degree of modularity, the way modules are laid out spatially, the way they interact across the lifespan from age 6 to age 85. Others have studied earlier developmental stages. I have a good colleague in the field who works even with in utero imaging and looks at the changes in networks that occur before birth. Very challenging, as you can imagine. But there is a thriving field of developmental connectomics and even lifespan connectomics now. The connectomic approach has been applied to anything you can think of. There's nothing left anymore to do that hasn't been done, at least in an exploratory study. Every possible disease you can imagine, every possible contrast has been run, including developmental processes. Because of issues with a lot of these studies being carried out 10 years ago with very small cohorts, we kind of have to go back and revisit some of this work again with larger cohorts that are becoming available to us because of larger NIH-funded consortia that are actually going through development, lifespan, as well as disease states, and make these data publicly available. And I think we have to, our methods have also become much more refined. One example is motion. Head motion is an insidious nuisance variable in functional MRI. The head is displaced sometimes very subtly by things like pulsatory motion in the vasculature, or breathing, or even yawning. And those displacements of the head in the scanner induce correlations that are unfortunately artifactual. Those have to be removed from the data. And even just the methodology for doing this properly is still evolving. And I suspect for a long time we will struggle with removing sources of noise and non-biological or unwanted biological noise from our data to get an ever more refined picture of what really is happening here. So this is not work that's been finished, we're done, we know everything about it. It's really just beginning, in my opinion. But I feel very strongly that without taking advantage of network approaches in this area, we have no chance, because our data are fundamentally about relationships and relational data in large systems, and network science is one of the most powerful methods that I know of to actually make sense of these data. We have focused on, a lot has been published in dynamic functional connectivity in the past 2 or 3 years. My initial slide was going to be an old simulation we've run in 2007 where we showed that there is dynamic functional connectivity. This was a computational model. As a computational modeler, you are not always met with respect by an empiricist. So we took our model to WashU, actually, big imaging center. I showed them how functional connectivity changes across time, and they looked at me like, we know it's stationary, man. There's nothing happening here. There can't be possibly. So now it's changing all of a sudden in the empirical studies. We kind of predicted it. One of the big things that we've been zooming in on is that there are characteristic fluctuations in modularity. So for me, modules and detecting communities is sort of something I've been drilling down a lot the past few years. We're seeing in human subjects that, in fact, there's individual differences in that are reproducible, that subjects go from high modularity states to low modularity states. Low modularity states, strangely enough, are closely aligned to the anatomy, so the functional connectivity is kind of sucked into the anatomical skeleton, and it's released when there are these high modularity states, when the functional connectivity runs free. In these high modularity states, we see a dissociation between task positive and task negative systems. Default mode, for instance, decouples from attention networks very characteristically. That's been shown by us, by Max Schein, Russ Poldrack at Stanford, and other groups now And we're intrigued by this. I think Mac and Russ are thinking of this in terms of neuromodulation, state changes in the brain that are triggered perhaps by the adrenergic system. We now find that these changes also occur in computational models that are lacking any kind of state-dependent mechanism, spontaneously, in other words. So we're trying to, a bit puzzled by that. We're trying to figure out what, whether we can explain what they've seen on the grounds of just spontaneous changes, no adrenergic system needed, or whether perhaps there's some additional things that need to be disclosed. So that's kind of where the story is from my perspective. But yes, there are dynamic changes in functional connectivity that are characteristic, that we can track, and that show interesting individual differences. And that's kind of where the field is right now.